This video is about phylogeny and ontogeny of immune system, evolution of innate immune system, and this particular study note is developed by APG Pathshala, MHRD team. So I would like to express my sincere gratitude to the development team of the study note. Thank you so much. Phylogeny and ontogeny of immune system, evolution of innate immune system. All forms of life possess some kind of defense system. Defense system needs to discriminate not only self from non-self but also defective, damaged and transformed self from normal self. Basic components of innate immunity are shared not only by a number of invertebrates but also by all vertebrates and rely on germline encoded receptors that are evolutionarily conserved as they target molecular patterns which are integral part of various pathogens. All cells of innate immunity express a large variety of PRRs or polyspecificity, and cells belonging to one class possess identical receptors. This is a striking difference between cells of the innate and adaptive immune branches, as in the latter, each lymphocyte bears a unique receptor. As all the cells binding a given microbial pattern are able to respond without the need for clonal expansion, innate effector response can occur very rapidly. Adaptive immunity has not replaced innate immunity but continues to be present along with and working in close association with innate branch to provide better protection to the organisms. Adaptive immune system requires signal from innate immunity for its activation. Building blocks of immune system are ancient and appeared and perfected at various stages during evolution due to selection pressure generated by dramatic environmental events on geological time scale, climate change and mass extinction. So let's have an introduction. All organisms are continuously exposed to pathogenic microorganisms like bacteria, virus, fungi and parasites. But because of the powerful defense mechanism, most of the organisms are capable of dealing with the outside of slide of pathogens. Defense mechanisms have been shown to occur in all forms of life, including bacteria, vertebrates and invertebrates. All these have a definite lifespan after which they die. Therefore, an equally important job that the immune system must perform is to dispose of dying cells to prevent unnecessary inflammation and maintain homeostasis. During the 20th century, pioneering discoveries by Julius Hoffmann, Bruce Butler, and Ralph Steinman, among others, led to the identification of the components of the immune system. Invertebrates have been shown to possess only the innate immunity, the first line of defense. That invertebrates make up more than 90% of all the Earth species itself suggests that even when considered primitive as compared to adaptive system, innate immunity is itself quite effective at providing adequate defense. A clear understanding of various aspects of immune system in different organisms might help researchers discover certain mediators which may prove useful for human therapies. Evolution of innate immunity. All forms of life have developed mechanisms to protect themselves from foreign intruders. Restriction enzymes and clustered regularly interspaced palindromic repeats or CRISPRs are able to degrade invading foreign pathogens in prokaryotes. As life evolved into more complex forms from unicellular eukaryotic cells to multicellular organisms, additional defense mechanisms arose to maintain cellular integrity and host survival. Various components of innate immunity already exist in the organism without prior exposure. Various components of innate immunity already exist in the organism without prior exposure to the pathogen, hence the name innate. Then cells of innate immunity. In 1882, Eli Marchinnikov was the first to observe aggregation of cells around an athon pushed into a starfish larva. He called the cells phagocytes. Phagocytosis first appeared in amoeba for acquiring food seems to have been retained in vertebrates and invertebrates where it may play a role in immune response. This property is due to cells which appear like macrophages and have been given different names in various groups such as amoebocytes, hemocytes, coelomocytes, granulocytes, monocytes, macrophages. Advanced invertebrates and cephalochordates and urochordates possess both granulocyte-like cells and macrophages. Increased diversity of cell types is encountered in jawless vertebrates and gnathostomes. Resident populations of macrophages are present in most tissues. Phagocytes, which functioned as digestive cells to begin with, retained their role to engulf the old dying cells as well as the foreign microbial invaders. It was the observation of Muchnikov that macrophages internalize apoptotic neutrophils at the end of inflammatory response that suggested a role for macrophages in the subsequent resolution of inflammation. In 1975, a killer lymphocyte was discovered which showed a rapid cytotoxic response against tumors and virus-infected cells without prior sensitization, and hence was named as natural killer cells by Eva Klein. This lymphocyte subset did not possess B and T cell receptors and was also referred to as a null cell. Thus, the natural question was asked, these cells function like CD8 plus T cells. Should they be part of adaptive immunity because they lack the receptors for T and B cells and respond to pathogen intruders among us almost instantaneously? Should be included in innate immunity? Though the issue has not been resolved, it has been demonstrated by Weisman et al. In 1997 that NK cells comprise a third lineage of lymphocytes being derived from the same lymphocyte progenitor as B and T cells. More populations of innate lymphocytes have been reported. NKT cells, gamma delta T cells, CD8 alpha alpha T cells, mucosa associated invariant T cells, B1 B cells, phagocytic with bactericidal abilities, and marginal zone B cells which require RAG recombinase for their development. Since the discovery of dendritic cells in the mice by Steinman and Cohen in 1973, not only a number of subsets have been described, but also their presence has been reported from all vertebrates. These cells typically show gene expression profiles such as TLR, MHC class II, B7 family, CD83, and CD209 DC. One study has reported that XL cells with long cytoplasmic processes from the spleen of Xenopus are capable of retaining unprocessed antigen at their plasma membrane and migrating into the white pulp following immunization. These XL cells have been proposed to be primitive follicular disease. As shown in many studies, follicular disease play an important role in humoral immune response by presenting native antigen trapped in antigen antibody complexes to B cells. These cells are of non-hematopoietic lineage, different from both macrophages and conventional disease. XL cells and Xenopus also express high levels of MHC class II and are capable of presenting antigen to T cells, thus indicating their role in both humoral and cell-mediated immune response. Disease expressing MHC class II have also been identified in thymus and skin of Xenopus, indicating that specialization of cells analogous to Langerhans cells occurred at the latest in amphibians. Other cells playing a role in the innate immunity are eosinophils, basophils, and mast cells. Leukocytes in teleost fish show morphological and functional similarity to mammalian macrophages, dendritic cells, neutrophils, monocytes, thrombocytes, and natural killer-like cells. However, presence of mast cells, basophils, and eosinophils has not been unequivocally reported in teleost fish. Eosinophils, basophils, and mast cells have been implicated in cellular responses to helminth infection. The dilution may have occurred to encounter metazoan parasites, worms, and parasitic arthropods, which are too large to be phagocytosed. Then receptor molecules of innate immunity. In order for the immune system to work efficiently in any organism, there is the need for discrimination between self and non-self, which is achieved with the help of receptors. Receptors of innate immunity are encoded in the germline. It includes a number of receptors. So even primitive organisms such as sponges show the presence of pattern recognition receptors or PRRs. These receptors have the specificity to bind pathogen-associated molecular patterns or PAMPs. This family of PRRs to be discovered was toll-like receptors TLRs. In 1966, Dr. J. Hoffman and P. Lamaitre discovered that Drosophila fruit flies bearing mutations in toll gene were very susceptible to lethal infection with Aspergillus fumigatus as compared to wild type flies. This observation of other studies indicate that toll protein is required for activation of innate immune response in invertebrates. In 1997, C. Janeway and R. Medsitov discovered a human gene for a protein similar to toll, which could activate the expression of genes of innate immunity in human cells. Therefore, this and other vertebrate toll relatives, which were discovered thereafter, were named toll-like receptors. In addition to TLRs, other PRRs were also discovered, as shown in the table, which may play an important role in innate immune defense mechanism. TLR toll-like receptors, NLR NOD-like receptors, then CLR C-type lectin receptor, RLR retinoic acid inducible gene 1-like receptors. Many invertebrate species contain very large families of innate receptors. For instance, sea urchin genome code for more than 200 TLRs, nearly 300 node-like receptors, and 180 scavenger receptors. Even though this diversity is less than that generated in adaptive immune response, the possibility of further somatic variation has not yet been explored. 7 TLRs have been identified in most primitive fish, iguanodonts, and 18 in bony fish, 14 in amphibians, 10 in birds, and 13 in mammals. Decrease in number of innate receptors in gnathostomes may have occurred due to the presence of adaptive immune system. C-type lectin receptor, CLR, is another family of cell surface RRRs. In humans, there are at least 15 CLRs, most of which recognize one or more specific sugar moieties such as mannose, fructose. Retinoic acid inducible gene 1-like receptor, RLRs, are soluble PRRs present in the cytoplasm of many cell types and functions as a sensors of infection by RNA viruses. These receptors are able to distinguish viral RNA from normal cellular RNA due to certain structural features. Viral RNA shows double-stranded regions, virus-specific sequence motifs, and RLR of RIG1 type identifies 5' triphosphate modification that arise during viral RNA synthesis and processing. Node-like receptors and nucleotide oligomerization domain leucine-rich repeat containing receptors, or NLRs, represent another family of PRRs. They are present in the cytoplasm, detect damage, and activate beneficial innate immune response. Human genome contains approximately 23 NLR genes, and mouse genome has about 34 NLR genes. Pattern recognition receptors, or PRRs, may be categorized in two ways based on their function. Signaling PRRs, TLRs and NLRs. Endocytic RRRs facilitate attachment, engulfment, and destruction of microorganisms by phagocytes without relaying an intracellular signal. Mannose receptors of macrophages, glucan receptors on all phagocytes, scavenger receptors that recognize charged ligands and mediate removal of apoptotic cells. Soluble pattern recognition proteins. These proteins bind to conserved repeated components on the surface of microbes. Once bound to microbe surface, these are recognized by membrane receptors on phagocytes, enhancing phagocytosis, and thus are called opsonins. Many soluble proteins function as opsonins. Surfactant protein A, SPA, surfactant protein D, SPD, mannose binding lectin, MBL, L-phycolin binds to acetylated sugars. Complement component C1q binds to bacterial cell wall components such as lipopolysaccharides. C-reactive protein CRP binds to phosphorylcholine and carbohydrate in bacteria, fungi, and parasites, as well as IgA antibody and some IgG subclasses. Although recognition regions of MBL, phycolins, and C1q have different specifications, their polymeric structures are quite similar, and so all are bound by CD91 opsonin receptors on phagocytes and promote phagocytosis of pathogen. CRP, IgG, and IgA bind to Fc receptors on phagocytes, facilitating phagocytosis of pathogen bound to them. It is important to note that microbes of quite different biochemical composition and with entirely different life cycles are recognized by rather similar mechanisms as by host PRRs. An equally important property of innate recognition system is that each class of pathogen is identified by a number of different PRRs to ensure rapid and potent inflammatory response. Lectins, a group of ubiquitous proteins, suggest their ancient origin, found in bacteria, plants, and vertebrates, bind to sugar molecules and cells, causing them to clump and get easily phagocytosed. Hemolymph binds to microbial surfaces, enhancing their removal, and RIG-like receptors for binding viral RNAs have also been reported from forms more primitive than the earliest non-vertebrates. All these represent pathogen recognition receptors, PRRs, capable of binding pathogen-associated molecular patterns, or PAMs. These molecular patterns are conserved among various pathogens such as virus, bacteria, fungi, protozoa, and helminthes. They are intrinsic to these microorganisms and necessary for their function and existence. These patterns are not found in host cells. All cells of innate immunity express a large variety of PRRs, polyspecificity, and cells belonging to one class possess identical receptors. This is a striking difference between cells in the innate and adaptive immune branches, as in the later, each lymphocyte bears a unique receptor. As all the cells binding a given microbial pattern are able to respond without the need for clonal expansion, innate effector response can occur very rapidly. An effector response is of innate immune system. Innate immune system works against infectious agents and individual tissues by antimicrobial peptides and proteins, producing antimicrobial peptides and proteins, phagocytosis, producing cytokines which play important role in inducing controlling acute inflammation, providing antiviral state, and stimulating adaptive immune system in vertebrates. Then epithelial barriers like skin and mucous epithelial layers protect the body's interior from the microbial invaders, not just by physical and mechanical means, but also by actively producing a great variety of antimicrobial peptides and proteins, or AMP. Some of the AMPs are lysozyme, lactoferrin, calprotectin, psoriasin, magainin, defensins, cathalycidins, and surfactant proteins, serum amyloid A (SAA) and serum amyloid P (SAP). AMPs, generally less than 100 amino acids long, present an ancient form of innate immunity as they are present in fungi, plants, invertebrates, and vertebrates. So, Hallmark et al. First characterized secropin, AMP. AMP in the sense nothing else, antimicrobial peptides and proteins, antimicrobial, from Cecropia moths. In Drosophila, 7 AMP families have been characterized such as attacins, cecropins, diptericins, drosomycins, deficins, drososins, and metnicovins, in addition to having a number of immune inducible proteins in the hemolymph and antimicrobial properties. Phagocytosis, nodule formation, encapsulation, cellular events, recognition by toll receptors, pattern recognition proteins, etc. Disease causing agents, breed integument, humoral events, antimicrobial peptide synthesis, prophenolactase cascade, lectins, and complement-like factors. Arthropods show various mechanisms to deal with parasites and pathogens. PRRs recognize each organisms either free in plasma or associated with various cell types. Phagocytes remove pathogens by phagocytosis. A sheath of cells removes a large number of microorganisms from the hemocoel by forming a nodule. So large invaders or damaged cell tissues are recognized and encapsulated by a multi-layer of hemocytes. So antimicrobial peptides and phenolphthalease may kill microbes, while lectins and complement-like molecules may play a role in recognition and elimination of pathogens. A number of reporters have been shown that unicellular parasites such as Plasmodium, Toxoplasma, Babesia, and Trypanosoma can be taken care of by host-derived deficits. So glands and frog skins secrete peptides called dermaceptins and magnanins, which have antimicrobial activity against bacteria, yeast, and protozoans. Then phagocytosis, even when strong defense mechanisms in place at the epithelial barriers, pathogens may gain entry into the interior due to wounds or insect bites. Next line of defense is made up by phagocytes. Phagocytosis is as old as the animal life itself. Upon insertion of a rose stone into a larval starfish, Mechnikov observed large phagocytes churning towards a foreign body and eventually forming a syncytium around it. A similar sequence of events occurs upon injury or infection to mammalian tissue. Studies of Mechnikov suggested that vertebrate leukocytes may have evolved from phagocytes of invertebrates. Bacterium phagocytosis receptors, phagocytosome, lysosome, phagolysosomes, soluble debris, exocytosis will be taking place. When microbial organisms bind to phagocytes via PRRs or opsonins, signaling pathways get activated, leading to polymerization of actin, allowing membrane to extend around and internalize the microbe forming phagosome. This is followed by fusion of phagosome with lysosome, and in neutrophils with performed primary and secondary granules, a number of antimicrobial agents attack and kill the pathogen. So phagolysosomes contain deficient catholicidins, low pH environment, a number of hydrolytic enzymes, and specialized enzymes to generate reactive oxygen species and reactive nitrogen species are shown by sponges, gastropods, fish, and several other organisms. So inflammatory response, in response to tissue damage and resulting infection, innate immunity induces a cascade of events called inflammatory response. A parallel sequence of events has been reported in the invertebrate inflammatory response. So invertebrate TNF-like molecules that show similarities to vertebrate TNF in size and activity. IL-6-like protein isolated from echinoderm coelomic fluid show physiochemical properties and activity similar to vertebrate IL-6. Biochemical studies on phagocytosis mechanism in echinoderm amoebocytes and vertebrate macrophages have revealed many functional homologies like microtubuli inhibitor cholecystokine, an inhibitor of microfilament organization. So cytochalasin B inhibit phagocytosis by both cycloheximide inhibitor of protein synthesis and fluoride inhibitor of glycosylases inhibit phagocytosis by both. So echinoderm coelomocytes could be activated to produce reactive nitrogen intermediate RNI in the presence of echinoderm IL-1, and the same way as the mammalian phagocytes show increased activity of nitrogen oxide synthetase upon activation. So cytokine-like molecules have been characterized from protozoum and deuterosome in invertebrates, tunicates and primitive vertebrates. Thus, as we go up to the phylogenetic ladder, we see evidence of a continuity of cytokines and conservation of inflammatory processes throughout evolution. IL-1, IL-2, IL-6, like that. IL-8 chemokine, IL-12 IFN, type 1 IFN, then TGF beta, TGF alpha, IL-15, IL-18, IL-4, IL-10. Okay, thus it is clear from this table that during vertebrate evolution, number of cytokines increased dramatically. Mammals show the largest number of cytokines, especially anti-inflammatory, in addition to the expanded network of cytokines controlling killer machinery of adaptive immunity. Selective pressures of autoimmunity and reproductive efficacy might have led to evolution of this pattern of cytokine repertoire. Fetoplacental unit in mammals represent a similar situation to a successful transplant. This is achieved by a specific cytokine network built up by NK cells, dendritic cells, lymphocytes, neutrophils, and macrophages, and other residual cells in the endometrium. Th2 cells and Th2 type of cytokines, IL-4, IL-6, and IL-10, predominate in normal decidua, inhibiting proliferation of Th1 and cytotoxic cells, Tc and NK cells. If Th1 cell activation occurs, there is release of pro-inflammatory cytokines such as IL-2, IL-1, IL-6, IL-12, IL-15, IL-18, and TNF-alpha and IFN gamma, which increase permeability of blood vessels, allowing access to immune cells which can reject the placenta as an allo transplant. Like NK cells, alpha delta T cells function in innate immunity as they recognize antigen directly, independently of MHC class 1 and class 2 molecules, but function in a specific fashion exhibiting memory. It has been proposed that alpha delta TCR-like receptor may have appeared before the rearranging antigen binding receptors. These cells are present in large numbers at the mucosal surface and mount immune response to various defective organisms entering via the oral respiratory route. Alpha delta T cells have been implicated in a number of functions such as secretion of a number of cytokines which can regulate function of other immune cells, providing help to naive B cells to produce IgM, IgT, and IgA antibodies, enabling macrophage recruitment and upon activation exhibits cytotoxic activity against many tumor cells using Fas ligand dependent and perforin granzyme or granulozyme dependent pathways. The complement system, a bridge between the innate and adaptive branches of immune system in vertebrates, have been postulated to have been rather very ancient. C3, a central component, and BF have been proposed to constitute the primitive complement system, having emerged more than 1300 MYA. They are similar to mammalian alternative pathway and have been retained by tetrodomes. Next evolutionary step of complement system occurred when chordates appeared with the recruitment of mannose binding lectin MBL associated serine protease or MASP and phycolin genes which established the lectin pathway. At this stage, lectins, an alternative pathway, might have played a role in opsonization and induction of inflammation. Final stage change involved complement gene duplication in the vertebrate lineage. Multiplication of C3 gave rise to C3, C4, C5 before the cartilaginous fish emerged. Since sharks have all the three genes, and such as those between BF, C2, and MASP C1RS, about 600 MYA. Before the appearance of cartilaginous fissions have probably contributed to classical pathway of complement activation. Components C6, C7, C8, C9 complement attack components might have appeared due to duplication of ancestral TCC genes before the jawed vertebrates appeared. Higher vertebrates host organisms show different pathways of macrophages function known as M1 and M2. In the M1 pathway, arginine can be converted to nitric oxide or NO by inducible nitric oxide synthase INOS, enabling macrophages to fight and kill pathogenic organisms. In the M2 pathway, also known as repair pathway, arginine is converted by arginase to ornithine and urea. Cytokines secreted by M1 and M2 cells are different. In vertebrates, macrophages have acquired MHC2 molecules, thus enabling them to play a role in adaptive immune response by antigen presentation to T lymphocytes. Then plasmacytoid dendritic cells, mainly found circulating in blood, have been shown to play a role in innate immunity by producing immune enhancing cytokines, interferons, which mobilize innate lymphocytes, NK, NKT, alpha, delta T, and help fight against viral and intracellular infections. As classical disease can capture and process the antigen effectively, migrate to lymphoid tissue and present the antigen to T lymphocytes for their activation. TCs bridge innate branch with adaptive branch of immunity. Mast cells, basophils, and eosinophils also play important role in the innate immunity. Eosinophils, mostly localized to mucosal surface, are able to respond rapidly to pathogens and bridge innate and adaptive immunity within secondary lymphoid tissue. They respond to helminth infections and allergens. Exposure of Drosophila to bacteria or fungi result in transcription of a number of genes resulting in antimicrobial peptides. Fungi and gram-positive bacteria lead to the activation of toll pathway, very similar to toll-like receptor pathway of mammals. Germ-negative bacteria activate IMD pathway, very similar to TNF receptor pathway of mammals. Hoffman, these studies suggest that there may be an evolutionary link between the regulations of antimicrobial peptide AMP gene expression in flies and mammalian innate immune response. Applications: controlling diseases in commercially important species. 25% of the global production of shrimp is lost due to disease. There is a need to understand the immune defenses of commercially important species. Viruses might have been responsible for the disease, and attempts have been made to develop vaccines against viral pathogens. They are suggestive of fast, specific, or primed immunity existing in insects and crustaceans, and it can be transferred from parents to offspring. Since many effector molecules seem to be shared by a large number of organisms, they might prove to be of therapeutic value. Then dermasiftins, isolated from the skin of South American frog, are polycationic peptides and inhibit a number of microbial cells including N. gonorrhea, Candida albicans, Plasmodium falciparum rapidly and effectively without any toxic effect on mammalian cells. So Limulus amoebocyte lysate provide the most sensitive and specific method for detection of microbial endotoxin in pharmaceuticals and drugs intended for human use. The complement of Alligator mississippiensis have been shown to have antiviral activity against human immunodeficiency virus type 1, West Nile virus, and herpes simplex virus. Various species of Naegleria and Acanthomoeba that have been found to be resistant to lysis by human complement show lysis by alligator complement mississippiensis. If parasites structure targeted by IgE are characterized, we may be able to identify homologous molecules and potential allergens in genetically modified organisms and new food items. Many studies point to the opposite possible use of alpha no delta gamma T cells for anti-tumor therapies, colon and pancreatic carcinoma, acute lymphoblastic leukemia, as well as antiviral HIV and HCV and antibacterial infections, tuberculosis and salmonellosis. Summary: in innate immunity, though non-specific and without memory, is present in both invertebrates and vertebrates. All cells of innate immunity express a large variety of PRRs, polyspecificity, and cells belonging to one class possess identical receptors. This is a striking difference between cells of the innate and adaptive immune branches, as in the later, each lymphocyte bears a unique receptor. As all the cells binding a given microbial pattern are able to respond without the need for clonal expansion, innate effector responses can occur very rapidly. It provides not only the first line of defense in containing the infection and conferring antiviral defense, but also stimulates adaptive immunity in jarred vertebrates. Research from a number of laboratories suggests the possibility of at least some of the molecules of adaptive immunity having arisen from structurally similar precursors serving related innate immune functions. For instance, complement cascade exemplifies gradual evolution of related molecules from innate to adaptive function and from non-immune to immune function. MHC molecules, though, play important role in adaptive active immunity by presenting antigenic fragments to T lymphocytes, also have many functions in innate immunity. From fish to mammals, we can see the presence of cytokines playing important roles in hematopoiesis. Inflammation and cytokine network seem to be conserved throughout vertebrates. During vertebrate evolution, number of cytokines increased dramatically. Mammals show a large number of cytokines, especially anti-inflammatory, in addition to the expanded network of cytokines controlling killer machinery of adaptive immunity. Selection test of autoimmunity and reproductive efficacy might have led to evolution of the pattern of cytokine repertoire. Comparative studies of innate immune system show different organisms provide us with many possible applications.